Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. And welcome and thank you, SynBYSS people, for joining us the 122nd seminar, and I call it a Stanford University Day. I am in Toulouse, France, where I just gave a talk at the SynBYSS conference with today's rising star speaker at the same session. Kate Adamala has been leading the Build a Cell webinar series for multiple years, and the SynBYSS conference is an annual in-person meeting like the inaugural SynBYSS conference in Hawaii this December. And I'm super excited and super happy to see European community and US community, of course, the global community working together to make this wonderful conference success. Many colleagues have given me feedback, inspiration, and ideas for our SynBYSS seminar series and the in-person meeting in Hawaii this year. I wanted to thank all of them for their invaluable and very, very priceless support and help. Okay, it is my tremendous honor to introduce very briefly, but probably that's impossible to do so, our pioneer speaker, Professor Stanley. He doesn't need any introduction because we all know him. By the way, he's a professor at Stanford University. I have known him for more than a decade since we were graduate students or postdoctoral researchers. His numerous papers pioneered the synthetic biology field, including his early cell paper on CRISPR interference. I believe many of you already use it. This CRISPR-I paper has inspired many researchers, as I mentioned earlier, including my lab people, allowing for gene expression control in a tunable, dynamic, and multiplex way. Now he changed the field a little bit, not entirely, but he's focused on more human kind of application using whatever tool he developed. And I'm excited to hear about what he's going to talk about today. Additionally, it has been a while since we met the last time, probably at the SEED conference, and I am really glad to catch up and listen to his talk today. Stanley, thanks so much for your amazing contribution to the scientific society, and the virtual podium is all yours now. And thank you again.
Great. Thank you so much, T-Sung, and for inviting me to this session. And I'm really happy to be also sitting on the same panel with Oskar and to share some high-level research that we've been doing. Should I go ahead to share my screen?
Please do so. Yeah, please.
All right. Will you be able to see anything?
Yeah, absolutely. Beautiful.
All right. Yeah, so for this audience, I know you all really interested in synthetic biology and how we can use synthetic biology for something helpful to the human. So for our research, we've been spending the past decade to think about how we can engineer the human epigenome to make better gene and cell therapy. I want to give you a brief update about doing. And I know everyone being very excited about the recent clinical approval of the first CRISPR drug named Casgevy to treat sickle cell anemia. It's one of the most, the first type of this drug, right, to modify the human DNA code as a permanent treat to treat a genetic disease that was originally had no real cure. This is a cure. So while very exciting, we also recognize there are some challenges that that's the future of the genetic disease treatment, because first look at how many of them there. There are more than 8,000 unknown genetic disease. While each of them could be rare disease, but they together affect of the global population. What do I mean by the challenge here? Well, unlike sickle cell anemia, which is very known for caused by a single mutation, many diseases are caused by multiple mutations, either affecting multiple genes or even affecting a single gene. This is a major challenge for gene therapy. For example, cirrhosis is a disease that affects the human lung. It has more than 2,000 known mutations that affect a single gene. Just imagine this is a rare disease, but each mutation affects about 100 people in the world. That really brings the question, for every single mutation, are we going to make a different gene editing modality by targeting a different location? Are we going to verify this efficacy specific and also prove they are clinical? Well, our thought is we should engineer more focus on the epigenetic layer for this type of disease. The reason is, for many diseases, no matter where the mutation occurs, the root cause of the disease is gain or loss of function of the genes. This really brings us to think about how can we engineer the epigenetics and allow the mutation agnostic therapy. For example, regardless of where the mutation occurs, we can always design something that binds to this gene, because this is caused by, for example, production of a toxic protein compound. So we can silence that protein and to remove this bad gene, regardless of the mutation. It's really brought some advantage, such as it's a single modality disease treatment, and to treat many patients, regardless of mutations. And furthermore, we believe, unlike the current gene editing therapy, the proposed epigenetic therapy has no DNA damage, thus it's much safer to use in human DNA. Well, that really speaks to an overview of what we try to do here. We try to edit the human epigenetics, not DNA codes. By epigenetics, what I mean is the chemical modifications that happens on the human chromatin DNA, such as people often talk about trying to methylate a DNA, thus the gene is permanently turned off, or demethylate a DNA to turn the gene on. This not only affecting that gene, but also affecting how long and the gene could be remain silenced or activated, all without changing the DNA codes. Well, that really speaks to what we have been doing 10 years ago, started with deactivated scissors. Like really, if you think about the Cas9 is a genetic scissor that people use for cutting a piece of DNA. We were the first to create a nucleoside decas molecule for nucleus deactivated Cas protein, and we introduced mutations to silence its DNA ability. And we further proved that this decas protein could pair with a guide RNA to target a DNA of interest. That really formed a basis for how we can use this decas protein to target a chromatin DNA, such as I have 20 base pair gene that affecting a disease that in the genome of cells. What we will do is we will design a decas protein, express them in the cells, and also pair that with a specific guide RNA. The guide RNA will carry some 20 base pair region, matching the 20 base pair disease region, and we co-express them, and they will survey the genome and sit on the target DNA to create a structure that is look like this. This is atomic force microscope from another group published, and which shows how the Cas9 guided by RNA binds to DNA. It forms a highly stable structure. And based on that, I'm going to show you something we can do to program to DNA as a basis for therapeutics. For example, we have devised different Cas9, Cas12, Cas mini, and each of them have a pairing guide RNA. When we deliver them into the non-dividing cell or dividing cell, they will create a DNA targeting system. And first, we can, based on system, by fusing them to transcriptional repressor or activator to silence or activate the gene expression of the target site. And second, we can couple them, fuse them to epigenetic enzymes, such as methyltransferase or to either silence or activate a target gene. Finally, we can also modulate the structure of the protein, such as the location of the DNA inside the nucleus or the interaction between the enhancers or the promoters to really control the structure. And very recently, we've been working on a system that is highly compact. We call search for small Cas molecules to better engineer deliver into cells for engineering the DNA in vivo. So the reason we do this is if everyone talk about Cas9, but Cas9 is a very large protein, and we desired much smaller protein. That's why we look at Cas12F, which is only a third size of Cas9. And when we look at this protein, we realize, great, it's a small Cas protein compared to the gold standard of the gene editing molecule. And we want to use them for epigenetic editing, right? But it almost have no efficiency in human genome. But its small size really guarantees the easy delivery. And the long story short, I'm not going to show you how we did it because the work was published. And we really did make a very efficient protein mutant come out from Cas12F, which is called Cas-mini. And that really solves this count here. It has really excellent efficiency. As my last final slide, and I want to show you how we can think about using such kind of small, compact Cas systems by remethylating DNA to target a disease such as called FSHD. It's facial scapulohumeral muscular dystrophy in collaboration with a company for preclinical clinical trial. So what we really talk about here is in the human muscle cells, in healthy people, there is a gene called DUX4, which is methylated. However, in patient, this gene becomes demethylated and they become expressed. The gene is toxic to the human muscle cells and causing the muscular degeneration. And since it's a very challenging gene to work with, and it's epigenetic disease, and also it's a relatively rare disease and have no cure at all. So we really think, okay, the DCAF system fused with a methyltransferase and with a guide RNA to bind specifically to the target gene and remethylate the gene to reduce its toxic expression. And with that, show that in human patient-derived muscle tissue cultures, we can use our system compared to a control, reduce more than 50% of the toxic gene production, which already clinically been believed that sufficient to like a rescue of the disease. We are very excited to move this therapy to human trials, hopefully in the next few years, to see how efficient and how safe it is. With this, I would like to really thank the opportunity from T.C. Wong to giving a talk about our work, and also I hope look for Oskar's talk. Thank you.
Fantastic. That is amazing. So I hope you save a lot of patient in near future by using your technology. So thank you so much for your talk. Okay, so now the main speaker of today with a longer introduction. Dr. Oskar Staufer received his bachelor and master degree from Heidelberg University. He went on to obtain a PhD in biophysics from the Max Planck Institute for Medical Research with the Professor Joachim Best, and then went on to do postdoctoral work in cellular immunology with Professor Michael Dustin at the University of Oxford. Since 2022, he is an Emmy Noether Research Group leader at the Leibniz Institute for the New Materials in Germany. There are too many German pronunciation I cannot do so well. His group combines synthetic cell approaches and biophysics to tackle key questions in immunotherapy. I believe I met him for the first time in person in Germany when I visited his institute twice, I think last year. Importantly, he is helping me as a committee member of the inaugural Synthetic Biology Conference, SynBYSS conference in Hawaii. Also, we are attending the same conference in Toulouse, France, the SynCell conference. Actually, we even gave, as I mentioned earlier, the talk at the same session together. So, Oscar, thanks so much for your time again today, especially for skipping the city tour time right now, other attendee actually going through, and your hospitality during my Germany visit last year twice. And please take it away. It's all yours now. Thank you.
Thank you very much. I hope you can see the screen. That should
Yeah.
work. Okay.
Yes.
Thank you very much, Moon, for the nice introduction, for the invitation to speak in your series here. I really appreciate what you've been doing for the synthetic cell community, also for the synthetic biology community in general. I went through the videos that you've been posting online. I think I'm one of the first to actually speak about synthetic cell engineering from the bottom up. That is a very growing and dynamic field at the moment. As you mentioned, we're on the same conference here, which takes place this year in Europe. Next year will be somewhere in the US. And I hope that with my presentation, I can show you some of our latest data on how we can build immunological systems from the bottom up by using synthetic cells. So maybe let me start by showing you one of my favorite movies of probably of all time, at least when it comes to immunology. It's a movie, unfortunately not recorded by myself, but by Alex Ritter and his lab. And you can see a human killer T cell in magenta here that is binding onto the surface of a huge cancer cell labeled in cyan. And the T cell is actually scanning the surface of this cancer cell to understand if it's good or bad. There is a lot of signal integration taking place on the molecular level, so biochemically, but also biophysically. And once the T cell reaches the decision that, well, this is a bad cell, it can actually even kill it. An amazingly complex process. We as a synthetic cell community, we've been trying to split vesicles apart for, well, roughly over a decade now. And then there's this thing happening in your body probably 1,000,000 times while I'm giving this talk, right? So it's just beautiful how immunology can work and how it can integrate so many complex signals. However, this process is also at the very heart of one of the most important medical revolutions of the last decades, which is personalized T-cell therapy. And this therapy, which I'm just going to try to summarize in 1 or 2 sentences, patient immune cells, cancer patient immune cells, are isolated from the blood and then brought into culture ex vivo for expansion. This expansion process is absolutely crucial. You need to start with some thousands of T cells that you were able to isolate and scale this up to several millions or billions to actually be able to genetically engineer them with chimeric receptors, for instance, so that when they're introduced back into the patient body, they will recognize the cancer and start to kill it. About 20 to 30% of all cancer patients can benefit from such a therapy. It can be actually healing the cancer in many patients. But as an engineer, you also see that 20 to 30%, there's still room for improvement, obviously. And there's different points where you might be able to improve this, maybe on the genetic engineering side, on the isolation. We believe that most of the potential has been missing in the actual second step, in the ex vivo expansion of these T cells. And to explain you why we reached this conclusion, I just want to briefly highlight how the industry standard for expanding T cells is currently looking like. The isolated T cells are brought in culture with stiff beads, magnetic beads, that have immobilized antibodies on their surface. These antibodies are able to cross-link specific receptors on the T cell surface, CD3 as part of the T cell receptor and CD28. And with some soluble cytokines around, this system is actually to very strongly and and very effectively activate the T cells. So they will undergo high expansion rates, incredibly high expansion rates, much higher than what you would see in vivo. Unfortunately, many of the cells that are generated in this way are of lower quality. That specifically means that they're very prone to immune suppression. So if they're reintroduced into the patient's body and they reach the tumor sites, or cancer cells are very well able to tune down their effector potency again. And obviously, this whole process, how it's happening now, does not reflect by almost any means how the in vivo process is happening. Well, how does the in vivo process look? Obviously, you don't have a bead in your lymph node and your spleen, wherever you get a T cell activation, you have an antigen presenting cell to start with. That antigen presenting cells have similar molecules on the surface, not antibodies, but effectively they activate the very same receptors. But that's obviously not all to it. That antigen presenting cell is much, much softer than a bead. And this softness and the biomechanical cues that the softness brings with it actually can activate downstream mechanosignaling within the T cells that contributes to the fine-tuning of the activation process. Additionally, there are cytokines exchanged in both directions that contribute a further level of regulation. And again, on the biophysical level, these receptors on the antigen-presenting site are laterally mobile and allow for a molecular self-assembly at the antigen-presenting cell T-cell interface. And that is absolutely crucial. Well, if we zoom out from that interface, there's additional context provided in your lymphatic organ. That might be, for instance, extracellular matrix that contributes to signal integration on the T-cell side. And then you have additional cells around that contribute and interfere into this activation process. So an immensely complex system. Many, many key players in there. And as a synthetic cell engineer, I see an opportunity in this. I see that with our tools of synthetic cell engineering that are specifically designed to tune down the complexity of such a system, we could contribute new means to actually improve the quality of T cells in ex vivo expansion models. So effectively, the aim of ours is to build ex vivo expansion systems for T cells that more closely mimic the critical steps of T cell activation in vivo to create multifunctional T cells that are better suited for immunotherapy. So what I'm going to present you is effectively data that follow 3 lines. One of our first questions that we raise, are we actually able to build 3-dimensional lymph nodes and lymph node environments based on synthetic cells outside of the body? Can we, with these tissues in hand, then maybe better understand on a fundamental level how biophysical and biochemical signals are integrated by T cells? And eventually we want to actually understand, can we apply such artificial lymph nodes or artificial tissues to enhance T cell therapies? Now, I talked a lot about synthetic cells at the moment, and in this community, it might not be obvious to each and everyone what is meant with synthetic cells, and many people in the field have different definitions of this. For us, a synthetic cell is any microscale structure that mimics either the function or the microanatomical features of a cell. And that can be on a very fundamental level, for instance, the cytoskeletal components, but in any case, it would be a compartment. And our favorite compartment that we developed are so-called droplet-supported lipid bilayers. These are synthetic cell models that are specifically tailored to mimic the biomechanical properties of an antigen-presenting cell, as well as the membrane properties of these antigen-presenting cells. As you saw in the previous cartoons, most of the signal integration happens at the membrane interface. So you want to be good at mimicking the membrane itself. So we produce microemulsions from simple oil and water emulsions with silicon oils. These are the advantage that they're viscoelastic, so they mimic quite closely the softness of these antigen-presenting cells. And onto the top of these microdroplets, we then coat a lipid bilayer onto which we can put antibodies or any kind of other ligands onto the surface. Let me go a little bit into the material science behind of this. You might ask, how is this even engineered in a high-throughput manner? Well, the emulsion process is pretty standard, a bulk emulsion to create such droplets. And then we stabilize the emulsions with a negatively charged surfactant layer. Onto that surfactant layer, we then deposit a layer of magnesium ions, divalent magnesium ions that give a positive charge to the whole thing. And this positive charge then actually acts like a glue for a negatively charged lipid bilayer onto which we can put then our recombinant proteins on the surface. So overall, these droplet-supported lipid bilayers are our working horse in the lab to actually study T cell activation. Let me give you a little bit of characterization of these synthetic cells. If we look at the membrane fluidity of the droplet-supported lipid bilayers by fluorescence recovery after photobleaching, we can see that, well, after the bleaching of the lipid bilayer, we have a fast recovery rate, so it's perfectly fluid. And if we look at the mechanical properties of these droplets by real-time deformation cytometry, we can see that we can, by, well, cross-linking the oil core chemically, vary the stiffness of these droplets over several orders of magnitude, either very close to physiological stiffnesses or anything that would be way above what you find in any antigen-presenting cell in your body. Okay, that was biomaterials-based characterization. You might now wonder what happens if you take these synthetic cells and put them in culture with human primary T cells. We put activating antibodies on the surface, and what you would see after some days, maybe even hours, is that the T cells will start to closely adhere and interact with these synthetic cells. If we measure the expansion rates of droplet-supported lipid bilayers with different stiffnesses, we see that we are almost as good as the industry standard, almost as good as the Dynabeats. Well, even though this is not statistically significant, just by looking through the microscope, you would see, okay, we are not as good, but we are still in a 50 to 80% range of what Dynabeats would achieve. However, if we look at one of the most important immunosuppressive markers, which is PD-1 expression on these T cells, we see that in all of our droplet-supported lipid bilayer models, we're considerably lower, almost only 1/4 of what you would get with Dynabeat-based expansion. And that is absolutely critical for good quality of T cells. You want them to express low levels of PD-1, but also maybe CTLA-4 and so on, so that once they're reintroduced into the patient and they reach the tumor site, they're not prone to immune suppression. A little bit more of phenotyping on this side now. If we looked at cytokine secretion of our droplet-supported lipid bilayer expanded T cells, we can see that critical regulatory cytokines such as IL-4 and IL-10 are heavily produced in these cells as compared to any Dynabeat-treated cells. And if we look on the surface marker expression level, I don't want to, put too many data of this, which is, you need to have a very solid immunological background to understand them. I'm just going to put exemplarily here CCR7 and CD62 ligand. We can see that they distinctly express these critical differentiation markers when expanded with our beads. Okay, that is perfect. I showed you now that by simply taking a soft particle that has a lipid layer on the surface and taking the very same antibodies that the industry standard use, we can get much better quality T cells. Well, we can do even better, I think. This is still a very dispersed system, while when we look at the lymph node, there is a whole microenvironment in there. It's a very specialized tissue that has different structures inside that guide the position and the activation dynamics of not only T cells, but B cells, and they interact with each other, positioning of dendritic cells, separation of CD4 and CD8, and so on. There's a lot of things happening. And in order to guide this process, also ex vivo, you need to actually to mimic this structure also in your culture. How do we do this? My PhD student, Anna, actually set out and took that challenge. Her first task to solve was, how do we go from a simpler unicellular dispersed cell system into a multicellular system? And how can we engineer such multicellular synthetic systems then to actually be a hybrid system so that it allows for the infiltration of human T cells that you can see here in green? Anna tested several conditions, and at the end, what she found out that is essentially the easiest and most elegant solution is by simply using a streptavidin-based cross-linking of biotinylated lipids that are embedded into the lipid bilayer of our droplet-supported lipid bilayers. So effectively, these lipids carry on their head group a little biotin molecule, and as streptavidin is a multivalent molecule, it can cross-link 2 or 3 lipid bilayers with each other. Okay, let's look at how this appears like. This is a stereo microscopy video of about 2 hours of our suspension of our droplets. And if I play this video, you can see that over time they start to settle on the bottom of the culture plate. The streptavidin starts to bind to individual droplets, and by a self-assembly process then forms macroscopic structures of tissue-like ensembles based on synthetic cells. Well, we can do this in a small scale. We can do it in a scale that you can actually see them by eye, and that they roughly match the size of actual lymph nodes that you would find in your body. Let me just highlight this here. Okay, how does it like on an ultrastructural level? If we look at the streptavidin, fluorescently coupled streptavidin, and the lipids in such a artificial tissue, we can see that the streptavidin accumulates at the connection sites of individual droplets and works as a essentially knob and hole technology to interconnect individual droplets. We can tune many things now. We can play with the biotin concentration. We can play with the streptavidin concentration droplets, and so on. For instance, if we play with the biotin concentration, in this case, it would be 1%. However, if we increase to 5%, you can already see that the typical anatomy of such a connection point would go more from a point-like structure to a planar structure. So the forces in between are much higher. And there's more streptavidin accumulating at the interface, and the area, as I said, is higher. And this also then translates to the macroscopic properties of such a tissue. For instance, if we compress our synthetic tissues and analyze the force that we need to induce a certain compression, you can see that we can tune this by simply adjusting the biotin concentration that we put on the membrane. Why is this important at all? I believe that most of the people that join the seminar or will see the recording might have experienced slight lymph node swelling when they got their COVID vaccines. That's a completely normal process if an antigen as delivered by the COVID vaccines arrives in the lymph nodes and starts to activate T cells, and these T cells will undergo proliferation, and the whole lymph node will start to swell a lot. The swelling also induces mechanical stress and so on. So effectively, the whole process of mechanical stiffening of that lymph node will contribute to the expansion process of the T cells within. So we need to be able to mimic this. Okay, now the most important thing that you might be asking is, what happens if we put human T cells in there? And to our very surprise, what happens is that these T cells will start to infiltrate the artificial tissue structure. They will start to crawl around and actually scan the surface of individual droplets. And if we put ligands on there, they will be activated. Actually, if we follow up on this several days later, now you can see here the individual T cell nuclei in cyan, we can find tiny proliferation spots within our tissue. And that is perfect for that moment, because we found that actually within our synthetic tissues, the T cells can proliferate, but still it doesn't add a real layer of control as compared to the dispersed system. You might be able to induce confinement effects and so on, but at the end, the limiting factor in this case is that the whole tissue is actually the same. It's composed of the very same droplets. However, if we look at the histology of a lymph node, we can see that's not isotropic at all. It's a very heterogeneous tissue structure, like every other tissue, that has specialized zones where either B cells or T cells would accumulate, and then have very specific functions for the differentiation and activation of T cells. So what Anna actually then did as a next step is she figured out a protocol to induce anisotropy or heterogeneity in our synthetic tissues, based on which we could, for instance, put T cell expansion zones in there, as well as scaffolding zones that are loosely interconnected and allow for a swelling process of the synthetic lymph node structure. Well, how did she do this? Well, she started with an isotropic tissue that was only composed of maybe 25% scaffolding structure, or increasing the droplets that actually would act as a scaffold to up to 75%. With a homogeneous mixture, and you get what you expect, a homogeneous distribution. However, if you, before putting both of these droplet types together, start the assembly process and mix and match them in a correct way, you can actually induce these anisotropic structures and induce heterogeneity in the system in a very controlled way. Does that have any impact on the T cells that would infiltrate in there? Well, yes, it does. If we look at the activating droplets here in magenta and the scaffolding droplets in green, we can see that the human T cells in cyan would preferentially accumulate in the magenta areas, so the areas that are rich in activating antigen-presenting cells. Yeah. And if we look on the functional level, this is now an example for the interferon gamma secretion of the T cells that we are cultivating in the synthetic tissues. We can see that the anisotropic tissues are much more efficient in activating and proliferating the T cells as measured by the interferon gamma secretion as the isotropic ones. And we don't exactly know at the moment why, but we believe that this is mostly related to the fact that anisotropic tissues allow for a better expansion of the whole structure when these T cells undergo the high proliferation phases. Let me briefly summarize this part of the talk. I showed you that we can produce synthetic cells that can be programmed to form tissue-like macrostructures. I showed you that this microautonomy patterns that we can produce help to vary biochemical signaling, in our case induced by antibodies, and biophysical properties of the T cells. I showed you amazingly, and we don't exactly know what's the driving force for that, that natural T cells will start to infiltrate these synthetic tissues. And maybe what was a little bit hidden in the immunological data, that the membrane fluidity that we put onto these synthetic cells is absolutely crucial to induce a therapy relevant and beneficial T cell phenotype that is less prone to immune suppression. Okay. That might be a very, very nice and straightforward way to expand therapeutic T cells ex vivo. But it has a significant drawback, at least at the moment, which is it's a completely static system. Any dynamics introduced in the synthetic tissues are introduced by the activity and the behavior of the T cells, while the synthetic cells are rather passive. And that is obviously not what happens in vivo. In vivo, the lymph node, the dendritic cells in the lymph node, the B cells in the lymph node change over time and in the time course of an inflammation. And that is not mirrored at all in our synthetic tissues so far. So we set out a second project where we actually tried to mimic the time dynamics of tissue inflammation, of lymph node inflammation, in a more controlled and sophisticated way. And for this, we relied on organ-on-a-chip technology. I believe in this community, I don't need to introduce too much about organ-on-a-chip. Essentially, these are microfluidic structures, mostly based on PDMS devices that allow to culture cells in a very controlled configuration, for instance, in the form of tiny channels. And by carefully selecting the structure of the cultivation device and the cell types and their sequence in which they're interfaced, and it allows to mimic quite closely physiological processes and anatomical structures that you would find in vivo. So our idea was, could we essentially apply this process to make our synthetic cells more dynamic? And we did a little trick for this. We relied on a sequential approach in that case. Instead of making the synthetic cells change their function, their structure, and mechanical properties, we decided to pass the T cells sequentially through individual modules that would represent the environment of a lymph node, either in the initial inflammation phase or in a very productive inflammation phase. This means that an initial module would have a different antigen load and different mechanical properties than an intermediate phase module that mimics more of a cytokine-rich environment, for instance, in an established inflammation, while the final module is more of a molecularly crowded environment and a cellular crowded environment that boosts T cell activation through confinement. This is how our chips look like. Essentially, our spirals where we pass on the synthetic the T cells one time or sequentially through each other. And actually, as a synthetic cell model, we had to change a little bit our approach. The droplet-supported lipid bilays that I showed you before are nice for these soft and dispersed cell culture systems, but they're not very resistant to flow and are especially hard to integrate into PDMS devices very stably. So we turned to silica colloidosomes. That is a very very well established synthetic cell module in the synthetic cell community. It's essentially a tiny shell of silicon nanoparticles that form a compartment that has an aqueous layer inside, and that allows to separate or to provide a surface for cellular cell culture. So how are synthetic organs on a chip produced? We start off with a simple 3D PDMS device based on an ABS negative that we remove the negative by acetone, and then we activate the surface with a siloxane treatment. In parallel to this, we produce our silica colloidosomes that, as I said, are essentially silicon nanoparticles that are mixed in an oil phase and then cross-linked as an emulsion by TMOS treatment to form these tiny compartments. And these are then introduced into the channels of our three-dimensional microfluidic devices. As the surface of these devices are activated, and the TMOS is present in the oil phase of the colloidosomes can start to bind to the surface of the channels. After this binding, we just need to remove the water phase, and we can introduce our T cells into them. Just to show you some images, how it looks like on the left is a magnified view of our culture vessel. In the middle of this slide, you can see the individual colloidosomes in a very packed configuration. And of course, we can tune the density of our synthetic cells are incorporated into these channels by simply varying either the TMOS concentration applied for cross-linking or by varying the total concentrations of colloidosomes injected into the channel. So we can create ones that are, very dense, so T cells would have a lot of interactions with the colloidosomes, or ones that have a large free area in the middle, so that T cells would mostly just being flushed through this. Okay. I said it's super important to have the lipid membrane on the surface, and the silica colloidosomes are essentially stable structures without any membrane mobility into their silica membrane. However, if we put lipids on the surface, again, we can reach the very same lipid layer mobility as we did with our droplet-supported lipid bilayers. That is a very simple process that allows for coupling of this membrane on the surface and then putting proteins on top, just as I showed you for the droplet-supported lipid bilayers. And amazingly, if, again, you flush human T cells through this, they will start to bind to the surface of these membrane-modified colloidosomes and crawl through the channels with this continuous medium flow behind them and scan again the surface of the individual colloidosomes. And if we quantify their activation by simply measuring the CD25 signal in the cultures, we can see that, again, if we couple anti-CD3 and anti-CD28 antibodies on the surface of the colloidosomes, we get high levels of activation. Okay, so far, nothing new. Having this aqueous volume in the core of the colloidosomes, however, is a distinct advantage to our droplets. The droplets have a hydrophobic environment, which is not compatible with many of the biomolecules that we would like to use. For instance, cytokines. Cytokines are highly important for the activation process of T cells. So we thought maybe we can provide us a further layer of control into the whole system by putting the individual cytokines into the aqueous lumen of the colloidosomes. In this case, a fluorescently modified version of the interleukin-2 molecule. You can see that while everything is in the oil phase, we can nicely encapsulate this inside, and once the whole thing is transferred into water, the colloidosomes would start to slowly secrete these interleukins into the extracellular medium. This is quantified here in the second part of the graph, but, and I now need to highlight the time scale of this release, this is about 6 hours. And if we talk about T cell expansion, you might guess by infection process that you've seen by yourself or witnessed yourself, this is a multi-day process. So going to 6 hours, which is our maximum release efficacy, doesn't really help us because it does not sustain the, let's say, 4 to 5 day incubation period that we want to achieve. So how we try to, well, I can take it now. How we solve this thing essentially is by integrating an alginate-based hydrogel within our synthetic cells that then provides for an additional adhesion of our interleukins to the hydrogel network inside, thereby slowing down the release kinetics of the interleukins. And this is exactly what happens. We can now go for a release dynamic that takes at least 3 days. And we can see from confocal microscopy images that the interleukin would bind to this intracellular, to the synthetic cellular environment inside. And this is what we think happens, and that slows down the release dynamics of the cytokines. It's very hard to do confocal microscopy in these channels because it's a three-dimensional microfluidic device. But you can see here a small layer of colloidosomes in the inside of a channel wall with the lipids labeled and the cytokine released into the channels, into the outside. Well, now to summarize the functional data, I'm just going to throw a lot of data around here, but what we did is then incubate human T cells in chips that have varying numbers of modules with different configurations, either rich in cytokines, low level of activating antibodies, or very crowded environment. And depending on what kind of configuration we choose, we can actually induce the differentiation of different types of immune cells, either some CD8 cells that are rather functional, so effector, or some that are more regulatory. You might now ask, because I highlighted in the first part so much that the quality of the T cells is much better. It's still the case. They're still express low levels of PD-1, so they're still less prone to immune suppression. And this, again, is based on the lateral mobility of the lipid bilayer on the colloidosome surface, and also based on the quite soft and a low Young's model of the colloidosomes in some 100 kilopascal range. Okay, with this, I would like to conclude the second part of my talk, in which I showed you that synthetic cells, such as colloidosomes, can be integrated as functional elements into microfluidic devices. I showed you that these synthetic cells can then interact functionally with immune cells cultured on chip. And by cleverly choosing the sequence of individual modules that the T cells would experience while they flow through the microfluidic devices, this allows a handle on the expansion process that mimics more closely the processes that actually happen in vivo. With this, I would like to thank all the people involved. Let me first highlight Anna and Niels, who have been really driving forces in the droplet-supported lipid bilayer part, as well as Nikki, who has put quite some work into the colloidosome and the three-dimensional synthetic organs on the chip. All the partners in the clinics and the biophysics departments around our institute that help us, the people from the Max Planck Center for Minimal Biology in Bristol, as well as my postdoctoral advisor, Michael Dustin, all the funding agencies that provided us with the support to actually do this research. And I'm looking forward to a lively discussion now. Thank you very much.
Amazing. I listened to summary portion, I guess, earlier yesterday, and then now I extended version, and I now understand better, even though I'm not expert in immunology. So let me ask some question, because I don't know the technical detail about immunology, so I'm kind of asking some high level kind of question based on your work. So I see huge potential out of your work, but I still see how difficult problem you are handling right now. So the question I want to ask is, to make this one really working in real practical application, I mean, translate this technology into real clinic, what kind of thing, you should address, or people collectively should address to make that happen?
So I think on the material side, the synthetic organ on the chip is more on a conceptual stage yet. I don't think we have any data to actually judge if on the translational potential of the whole concept.
Right. The first part of the talk.
Yeah, for the first part, however, there are some, I would say, material science improvements that we need to do. We need to work a little bit on the standards in terms of how we produce the emulsions. The materials that we use are absolutely fine for the clinics. It's standard lipids, approved oils. The magnesium doesn't play a role, and the antibodies we use are not special at all, and would be subjected to the same regulatory mechanisms as any antibody. What we need on the biological side is a much better understanding of the T cell phenotype that we're actually inducing. I showed you that are less prone to immune suppression. They seem to have slightly distant functions, but sooner or later we would need to test this in more therapeutically relevant models, for instance, in a CAR T cell model, in a bispecific T cell engager model, and so on, to get a better understanding where are the limitations of these T cells, but also where are potential advantages as compared to the standard in the field. Measuring immunology in vitro is I think, a somewhat okayish predictor to what will happen in therapeutic context, but sooner or later to actually show the value of an approach for immunology and specifically for immunotherapy and in vivo context needs to be studied.
Got it. Let me check whether there is any question from audience. Okay. Let me ask some kind of question for the junior. So I'm talking about student and PhD student or master or the postdoc wanting to do potentially immunology research. The reason I'm asking that question is when I was the master student, it's, oh my God, it's almost 30 years ago. I did want to study immunology, so I literally bought immunology textbook. I went through all the way to half, and then when I reached the half of the textbook, I realized it's too difficult for me to, work on as of my whatever project, because at the time I working on microbial system to make this called the FK506. That's an immunosuppressant molecule, out of the bacteria fermentation. So I want to understand, but I realized it's too much for me. So if you wanted to give some advice for the younger student or postdoc who really want to do immunology, because now everyone somehow understanding because of COVID, right? What would be your advice to them?
It's interesting because I'm not a trained immunologist myself, right? I did PhD in biophysics, but then I did some immunological work in my postdoc. So I did this transition and this process of getting into immunology quite recently. And for me, I think at the moment, the most important thing is not to get lost in too much data, because immunology is a field where there's tons of data around, but really try to focus on understanding specific principles on the cellular level. Molecularly, there are tons of things happening, yeah. But sooner or later, you will need to interface the problem of the data and the quantity of data. For me, what helped the most is to look at the one very selected and specific process, which was the activation of CD8 T cells with a specific type of bispecific T cell engagers. So very, very narrow. And from this, once you understood that tiny process, you can work your way out and understand what all the connections coming from different areas of immunology. That would be my advice. Start to get a general overview, then pick out one specific problem and work your way up from that very, low-level technical detail problem.
Got it. Because what I did is kind of reading the textbook, and that is somehow make me sleepy entire time, because I'm not doing real world problem solving at the time, just reading something very difficult, textbook or somehow to me. So probably that's why I didn't pursue that way. Okay, I got a question from audience. As an application, what would the process for this therapy look like, starting from a known antigen of interest?
So in a technology transfer approach, our synthetic cells would come in the second step in the ex vivo expansion process, and we would be using most likely a very specific antigen as the one asking the question suggests, right? And that would need to be defined based on the therapeutic context. This peptide would be loaded onto an MHC molecule that is immobilized onto the surface of the droplets. And then the T cells from patients would be incubated with our droplets. This activation happens right? That is very antigen specific now and would give you probably a very, very small set of T cells that are activated because, well, less than 1% of T cells isolated would be specific or show any type of specificity to that particular antigen. So this is why this type of expansion process is usually not performed. What would usually happen is a very unspecific expansion of T cells. Yeah, so we would take our droplets and just activate all the T cells that are isolated, expand them, and then there would be subjected to a transgenic modification based on genetic engineering with antigen-specific CAR, chimeric antigen receptor. And this is how you would introduce antigen specificity into the expanded T cells.
Okay, so thank you. Now I see, okay, I have to see another question. That is, would incubation happen in the chip? Is the artificial heterogeneous structure, or are the droplets themselves enough?
So enough depends on what your aim is in a therapeutic context, I would say. Yeah, for the moment, we believe we are already better than the industry standard. Yeah? But obviously, it would be the nicest approach to introduce a dynamic behavior in our synthetic tissues in the first part of the talk, right? For instance, any light-guided or magnetic field-guided changes in the tissue structure, any in vitro transcription translation-based secretion and production of proteins that contribute at distinct stages of the T cell activation and tune the process, right? The on-chip incubation, I think, could be most valuable for people doing immunological research, right? That want to find out in a discovery-driven manner how or in which sequence specific events need to take place for a specific type of T cell being produced, right? I see this more of an understanding approach to immunology.
Got it. Okay, let me double check one more. Okay, so that's all our question, and it's perfectly 11 o'clock Central Time. So why don't we just close? Okay, so thank you all for joining and staying today. We'll meet again on April 25, Thursday, the same time, the same Zoom link. We'll have the Dr. Barbara Giordantana, a director for European Innovation Council funding programs, and also Professor Katie Galloway at MIT. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. Thanks. I stop recording. Just give me 1 second.